Before we begin the presentation, I would like to introduce today's speakers, Drs. Venkateswari, Muthukrishnan, and Nikunj Patel. Full speaker bios can be found in the blue widget at the bottom right of your console. Aswari is a senior scientific consultant at Certara. She is responsible for training and pre-sale support in the South Asia Pacific and the Middle East. She provides training and support for a number of Phoenix products, including Phoenix Win Non-Lin, Phoenix NLME, Phoenix Connect, and the IVIVC toolkit for Phoenix Win Non-Lin. Prior to joining Certara, Aswari held positions at a number of pharmaceutical and consulting companies. Nikunj is a senior research scientist in Certara's modeling and simulation group. He leads oral and dermal absorption projects and is a member of the cardiac safety simulator development team. Prior to joining Certara, Nikunj worked at Tata Consultancy Services as a research scientist. Nikunj and Aswari, welcome to today's event. I'll now turn it over to Aswari to begin today's presentation.
Thank you, Susan, for the introduction. In today's webinar, we briefly discuss about various applications of IVIVC models during drug development as well as during the post-marketing. So we will introduce conventional methods that involve deconvolution using Wagner Nelson, Loo-Riegelman, and numerical deconvolution, and then DTC, the modified deconvolution as a part of IVIVC toolkit. Next, we also discuss about the advantages and limitations of these conventional methods along with the mechanistic methods. So we will introduce the concept of mechanistic IVIVC, which is based on physiologically-based pharmacokinetic modeling. While mechanistic models require more data and sophisticated software, they can overcome some of the limitations of the conventional methods, especially for the molecules that involve complex ADME processes. Finally, we will present two case studies involving Metoprolol and ER-Diltiazem, where we are going to discuss about how various methods that we presented, conventional as well as mechanistic methods, are used in these examples. In the end, the suitability of either conventional or mechanistic model will be discussed. Now, what is an IVIVC? An IVIVC is a mathematical relationship that is predictive of a pharmacokinetic parameters such as Cmax and AUC from in vitro dissolution data. Drug developers frequently use IVIVCs to serve as a surrogate for human bioequivalence studies and to obtain biowaivers. It is also used to support and validate the use of dissolution methods and specifications. In addition, it is used to assist quality control during manufacturing, and finally for selecting the optimal formulation by the formulation team. Now we will take a poll question on IVIVC to understand the trends in the industry. If you would like to participate, please do so at this time. So it looks like most of the industry is using IVIVC models to support formulation development. However, a percentage of pharmaceutical industry also using it for obtaining biowaivers and to support the dissolution specifications. Now let's move on to the key component that differentiates between conventional approach and mechanistic approach, that is deconvolution. So let's look at how to develop an IVIVC model. So IVIVC models are developed using in vitro dissolution data, that is mean in vitro dissolution data, and by getting the plasma concentration time data from a crossover study after administering all of the formulations to be used as internal formulations in the IVIVC model development. In addition, a reference formulation is administered either as an oral solution or IV bolus or an immediate release formulation. So why do we obtain the in vivo data? So we use the in vivo data to deconvolve and obtain input that is fraction absorbed. So finally, we try to correlate in vitro dissolution data with in vivo fraction absorbed. So next, what is the deconvolution? Let's look at it closely, what is involved in it. So the deconvolution is defined as response is a product of input and system behavior. So in our case, response is the plasma concentrations that we have obtained, and system behavior is how the human body processes the drug, that is disposition. This is unique to a molecule in a given species. So now we use response and system behavior and we obtain input, that is fraction input. So during this process, how we deconvolute, and then quality of the deconvolution depends upon how we define the system and then parameterize it. Now let's look at various methods of, conventional methods of that are being used for estimating the fraction absorbed. Wagner Nelson method. This method assumes human body as one compartment, and it is not appropriate for the drugs that follow multiple compartmental characteristics. No assumptions of zero or first order absorptions are made. It doesn't require IV data and gives the fraction absorbed. So while the limitation of multiple compartmental characteristics are addressed with Loo-Riegelman method, because it will address estimate the fraction absorbed for the molecules follow more than 2 or more compartments. However, it requires both extravascular and intravenous data from the same subject. These two methods cannot be used for the nonlinear elimination. They deconvolute the whole systemic input rate, the total fraction absorbed, through measuring the plasma concentration, which is a composite function of in vivo dissolution, GI transit, and permeation and first pass, etc. Next we will look at the most commonly used method, that is numerical deconvolution method. In this method, there is no model assumed about the number of compartments or the kinetics of the absorption. It requires both extravascular and reference data. The reference data can be oral solution or immediate release formulation or IV data, which is very convenient. Most often when the formulation or when the API is developed for a oral formulation, they may not have tested that as IV route of administration. So that's why it is more commonly used. However, it makes a lot of assumptions such as linear distribution, elimination, and time invariance, meaning the input site is the same for all the formulations, and then the input rate between the two time points assumed to be constant. It may not be the case. So depending on the UAR used, that is, it is a single exponent or multi-exponent, meaning one compartment or two compartment exponents. It deconvolutes a composite function of in vivo dissolution, GI transit, permeation, first pass, etc. So you will not get separate in vivo dissolution data. However, it is simpler and sufficient for IVIVC model where no complex ADME processes are involved. Now I will hand over the presentation to my colleague Nikunj. So he will present more on mechanistic IVIVC approach.
Yeah, okay. Thank you, Ashwari. It was a nice introduction to IVIVC and conventional methods. So the main difference between numerical and other methods with mechanistic is that with mechanistic method, you can separately estimate or different processes that lead to an overall systemic absorption of the drug, that is including dissolution, gastric transit time, permeation, gut wall metabolism, and first pass. So with this mechanistic model, you will get different processes and also the final outcome, which is nothing but the drug leading into systemic circulation. So how it can improve the chances of IVIVC? For example, this is a plasma concentration profile. With mechanistic method, you can deconvolute the yellow line, which is in vivo dissolution. Then blue line is the absorption into enterocyte, and the red line is the absorption into the blood. So the dissolution starts probably from time 0 if there is no lag time, but then the absorption can only occur when the drug is emptied from stomach, so there is a delay. And basically not all the drug that is dissolved gets absorbed, because some of them might be escaping the absorptive area of small intestine and colon and maybe leading into feces. Also, there will be a first pass elimination, so the maximum amount which is reaching systemic circulation may be much lower than the amount dissolved. Similarly, with conventional methods, this is considered as a maximum and 100%, and then the whole fraction is recalculated. So, for example, the in vitro dissolution is in vitro property is mainly dissolution. So when we try to correlate the green line with the yellow, there might be a simpler IVIVC because then this is dissolution versus dissolution. But when we probably try to correlate the dissolution with fraction absorbed, we might need a complex IVIVC function because we are not correlating the two same properties. So that is the main difference, and with that we can, it leads to our next question, poll question, which is what is the current approach that you use at your organization to perform IVIVC? So please fill in your response to the poll. Okay, so I guess you submitted your response. Let's look at the results. Okay, so about majority of the people use conventional IVIVC, and I think that will probably be numerical because that is a gold standard. There is reasonable amount of people using mechanistic IVIVC and also a good amount of people using both approaches. So I think both approaches and conventional are the leading responses. However, there are also few people who are not using IVIVC, and probably we would be able to explore more. Why not? I think with this, I will probably pass it back to Aishwari, who will take us through the process of developing IVIVC with Phoenix Toolkit and leading to case studies. So, Aishwari.
Yeah, thank you, Nikunj. Now we are switching gears. So now we are going to discuss about the steps that are involved in IVIVC model development with specific to IVIVC toolkit. So the first, we will look at the molecule API and try to understand whether what is the likelihood of IVIVC is likely to succeed or fail based on the classification, whether it is DCS class 1 or BDDCS class 1 or class 2. So once we have the probability that a molecule API belongs to this category, it's highly likely we'll be able to develop a IVIVC. The next step is to understand the data requirements because we need to generate appropriate data to develop an IVIVC. So for that, so you would like to have in vitro data from all the formulations. Ideally, you should have fast, intermediate, and then slow release formulations. So that is recommended. If your dissolution media have to be, discriminatory, at least 10% difference between the formulations. Once you have in vitro data, you need to have the corresponding in vivo data in a crossover study. In addition to that, you need to have the data corresponds to either oral solution or immediate release formulation to estimate the disposition function or UAR. That's what the terminology used in IVIVC toolkit. So once you have this data available, then you'll start with modeling the dissolution data. So you take the mean dissolution data and try to fit one of the built-in models available in the toolkit, that is either Hill, Weibull, or Makayed-Banakar, or double Weibull models. So you have the data fit to one of these models, so you would be able to predict dissolution at any given time point. Remember, to seek a biowaiver, we need to establish a level A IVIVC. That meaning point-to-point correlation. So the in vitro dissolution data at a given time point has to be matched with in vivo dissolution data at another time point. So once you have that predicted dissolution data based on the model, then the next step would be to look at how to get fraction absorbed. So one of the first steps in the fraction absorbed estimation is getting the UAR from the reference formulation. So the reference formulation, if it is an oral solution, we have to make sure that we remove the absorption component from the whole profile. So you fit the data and then you only look at the disposition function and then use it as a UAR. So you use the UAR and then deconvolve along with the plasma concentration data and then get fraction absorbed. Once you have fraction absorbed and then fraction dissolved data, we will be able to generate two plots that are useful to determine which of the IVIVC models are more appropriate. So those plots are fraction absorbed versus fraction dissolved, or Levy plot, which is T vivo versus T vitro, that corresponds to the same percentage of drug absorbed or dissolved. So now when we have to decide about what are the factors that needs to be accounted in the IVIVC model, so they can be T-scale. If you have time scale differences between vitro data and T vivo data, then you would have to apply T-scale, so the difference between the slopes. T-shift is the parameter that accounts for the lag time in vivo data. Fabs scale is another parameter. If you have differences between the overall bioavailability between the formulations, so you would want to account for the Fabs scale. Fabs base is not very frequently used. It is only applicable for the formulations which release endogenous. Maybe an example, corticosteroid. So you have some cortisol, you may want to account for the Fabs base and subtract the Fabs base in your IVIVC model. So X equal to Y or fraction absorbed equal to fraction dissolved is the simplest IVIVC model that you can think of. But most of these built-in models that are available in IVIVC toolkit, they account for the Fabs scale and then T-scale, and then in addition, the next model is account for the lag time also. Finally, the last model was accounting for the endogenous concentration by including the parameter Fabs. So now once you have the IVIVC model developed, the next step is to predict the concentration using the model, and then you have predicted plasma concentrations and then observed plasma concentrations. Then you would be able to estimate percentage prediction error. So if you wanted to validate a level A IVIVC model, level A correlation, then you need to have percentage prediction error for all the formulations. The average should be less than 10%. One of the formulations can be less than 15%. So if it is more than 15%, and then it can be inconclusive. And for narrow therapeutic drugs, you may have to use external validation too. So now let's look at closely about the plots we discussed, that is fraction absorbed versus fraction dissolved. So as you see this plot, if your data points that are deviating from the regression line in the end, that means perhaps you may have to account for the Fabs scale. So the model should include Fabs scale. And Fabs base, if you look at this and then see whether do you see any intercept on y-axis, perhaps you may have to include Fabs base. Moving on to the Levy plot, which is T vivo versus T vitro. So where you see here, if your regression line is deviating from the line of unity, that means you have differences in the time scale in T vivo versus T vitro. So you need to account for by incorporating the T scale, so which is accounting for the slope differences. T shift, if your regression line is intercepting somewhere on the x-axis, so you have a lag time in the absorption, so you need to include the parameter T-shift. So next, once you have examined these two plots, then you can decide which correlation model is more appropriate. Now moving on to the case studies, we will discuss about various approaches used. We also include the literature published data, and then the same data is digitized and used. So just to look at the background of this case study. So this case study is coming from Eddington et al. 1998 publication. So where they have tested metoprolol controlled release products. So as you know, metoprolol is a BCS1 and BDDCS classification 1. So the objective was to develop and validate an IVIVC model. This study included 3 multiparticulate extended release formulations, fast, moderate, and slow. So the resistance data used was oral solution. In addition, they tested 2 types of dissolution apparatus, and they found apparatus 2 is more appropriate and more discriminatory. And in vivo data was generated from 7 subjects in a crossover design. Now look at the published data on the metoprolol. So in this, the in vitro data was fit to a Hill model and for the cumulative dissolution, and then in vivo data was developed or estimated using the numerical deconvolution methods. So the PK parameters, Cmax and AUC, were estimated using Phoenix Win-Nolin. As you see here, while the R-square value is 0.95, you still see there are points that are far from the regression line and are not able to be very well explained for, from this IVIVC plot. And then you can clearly see that by looking at this x-intercept, there is a lag time. Okay. However, if you look at the percentage prediction error, the average is 3.5, which is less than 10, and then any one of these formulations are all less than 10, suggesting that you have a robust model that qualifies for the validated IVIVC model. So, taking the same data, digitized, and then put it into IVIVC toolkit, then the first step, we would like to make sure that the rank order for the in vitro data and then rank order for the in vivo data matched. Basically, the fast release formulation is giving the peak faster versus compared to the slow release formulation, the peak is much more prolonged. So the oral solution is 50 milligrams and then formulations corresponds to 100 milligram dose. Okay, so with this, when we develop the IVIVC model using the deconvolution method, the fraction absorbed is equivalent to upscale multiplied by fraction dissolved, accounted for the t-scale multiplied by t vivo, and then also you have a parameter that accounts for the lag time. So you see it now, the regression line is going through the zero, and then most of the data is close to the regression line. And then you look at this precision of these parameter estimates for upscale, t-scale, and t-shift are reasonable. So in addition to that, if you look at the percentage prediction error, they are also less than 10%, suggesting that the model is qualified. So when you look at this model compared to the published data, so this model accounted for several things, the lag time, and then able to address some of these dispersion in the earlier time points for the fast dissolving formulation. However, let's look at the mechanistic approach for the same formulation. Is estimation of the in vivo dissolution is better than fraction absorbed. And by understanding this mechanism, it would help the formulation scientists. Maybe you should consider in vivo dissolution during formulation development. You would better understand the mechanism. So now I will let Nikunj discuss about the mechanistic model results.
Yeah, thank you, Ishwari. So I hope you can see the slide. It's small figures. I hope everybody is able to read the font. So the first image is that the same data that Ishwari mentioned, we subjected it to the mechanistic model, mechanistic IVIVC, which is in the SimSip simulator, and we estimated the in vivo dissolution. And then you can see the correlation. So the plot A on the left top corner is fraction dissolved estimated from mechanistic IVIVC and correlated with dissolution. So you can see there is a very robust correlation, no lag time, because the lag time, which is probably because of the gastric emptying, is already accounted for in the model. And also all the formulations are spread uniquely around the line, so there is no bias in the IVIVC. In comparison, this is another plot which was published in 2002 by Sirisut and Eddington. So they used a semi-mechanistic approach to account or to separate the first pass elimination from the dissolution so that they can get some better estimate of the dissolution separately from first pass. So maybe they were able to account for the first pass, but still there was bias for the fast release formulation. So the slow and medium release formulations are uniquely surrounding the line of IVIVC, but the fast release formulation is clearly separated. This is the original publication of Eddington et al. So you can see there are some points which are deviating from the line. We cannot know whether these time points belong to fast release formulation because that was not clearly available in the publication, but I think this is also fast release formulation. So generally, it is an assumption that when people use solution data as a unit impulse response, people should get pure dissolution as a deconvoluted profile, but this is clearly not the case here. There is a lag time as well as there is a deviation. We subjected the same data in the IVIVC package and also estimated and deconvoluted the data. So you can see that the same bias for fast release formulation also exists in the numerical deconvolution method of our IVIVC package. So we wanted to understand what's exactly happening. So we also, in mechanistic model, you can estimate the dissolution profile as well as the absorption profile. So this is a dissolution versus dissolution from physiologically-based method, and this is a fraction absorbed from the physiologically-based method correlated with dissolution. And you can see that there is now a bias for the fast release formulation. So basically, you can say that the PBPK also is estimating the absorption, and when we try to correlate absorption with dissolution, there is a bias. However, when the same data was subjected in the Phoenix IVIVC toolkit with those T-shift absorption scale and T-scale parameters, you can see that empirically the model is able to account for the lag time as well as the bias towards fast release formulation is reduced. So basically, with mechanistic model, you can separately account for the mechanisms and then estimate a pure dissolution, or you can use a simpler model with empirical parameters like absorption scale, T-scale, etc., and still you can have a nice, robust IVIVC. Now, as Ashwari mentioned, with mechanistic model, you can try to understand the sources or the reasons why there was a bias for the fast release formulation. So these three are the same plots we saw on the last slide. And you can see that there was a bias for the fast release formulation for the these conventional two methods. And when we tried to look from the mechanistic model, different profiles, so the green dots is the dissolution, in vivo dissolution versus in vitro dissolution. The purple line is the fraction absorbed into the gut wall, so that is the fraction permeated, and the blue line is the fraction reaching systemic circulation. So you can see that metoprolol is a high first pass extraction drug, so only about 50% of the absorbed drug reaches systemic circulation. And for the fast release formulation, a significant proportion of the dissolution profile is controlled by gastric emptying, because if you look at the dissolution profile of fast release formulation, it releases almost 80% of the drug in less than 2 hour time. So most of the dissolution and release is happening in stomach. So the absorption is majorly controlled by the gastric emptying rather than the dissolution. So when the rate limiting step is a gastric emptying, if you try to correlate deconvoluted data with in vitro property, which is dissolution, there is going to be a bias, because the profile is, the absorption is not significantly controlled by dissolution but by gastric emptying. However, the impact of gastric emptying is much smaller for medium release and slow release formulation. So for this formulation, there is minimal bias with using numerical method. So that is explanation for the that is probably a plausible explanation for what is happening with this metoprolol formulations and why there is a bias in pure numerical deconvolution. With this, I will pass it back to Ashwari, who will take us through the second case study. Ashwari?
Yeah, thank you. So now the second case study includes Diltiazem. So it is a BCS class 1, and then it has high first pass. So the data was extracted from Fdiss et al. 2002 publication. So the goals were very similar to Metoprolol: develop and validate an IVIVC model for Diltiazem formulation. So there are 3 multiparticulate extended release formulations: fast, moderate, and slow. So the in vitro data was again tested in 2 apparatus, USP apparatus 2 and then 3 at different speeds. However, the apparatus 2 with the pure distilled water found to be suitable. Then in vivo data was generated. In addition to that, oral solution was used for the reference formulation. Now let's start with the published data for this diltiazem. So as you see here, the authors have used a quadratic equation, which is a nonlinear equation. And then if you look at the average percentage prediction errors were very high. So when you fit the data into numerical deconvolution with only a couple of them, so the percentage prediction errors were dropped. So if you include all three formulations, then you would have high percentage prediction errors. However, if you only include medium and fast, then you are able to reduce the percentage error. Still, it is higher, and the model did not qualify for the validation. So the others attributed for this high percentage prediction error is due to high inter-individual variability due to first pass metabolism. And then you also see here the slow release formulation and then fast release formulation are showing some bias. Let's move on to the Phoenix IVIVC toolkit. So we first started examining the plots for in vitro and in vivo. As you see here, for the fast release and intermediate and then slow release, in fact, there is a lag time in the slow release. So there is a scope for improvement in the dissolution medium. So however, you also see in vivo there is a lag time, but it is not as prominent as for the slow release formulation in vitro data. So we used the oral solution to get the UAR estimation, and then we have used the same model that accounts for the app scale, and then T scale, and then T shift. However, in this case of diltiazem, as you look at the CV %, which is a precision, indicator, there is a high uncertainty for the T shift parameter, while app scale and T scale seems to be better. So I have tried the other model without T shift; it was not better. So now, in this, when you look at this plot, the lag time is accounted for. However, the slow release formulation, there is a higher absorption. That could be just because you have higher or prolonged lag time in vitro data when compared to in vivo data. So when you look at the percentage prediction errors, so in fact, it did qualify, but you see here for the fast formulation, it's higher than 10%. However, it should be less than 15%. So the model is qualified, but we do see a bias for the slow release formulation that may be better explained by using the mechanistic approach, because the diltiazem is a complex molecule overall. So I think I will give the presentation to Nikunj.
Yeah, thank you, Eshwari. So, okay, in the same data, so here in the same data was subjected for in vivo dissolution estimation in SimSip IVIVC, which is using PBPK framework. And in this slide, I think all the three results are shown in the same slide for comparison. So this is the original data, where they used conventional numerical deconvolution or the old numerical deconvolution method. And you can see that there was a very widespread around IVIVC, which probably indicates that there is going to be a higher prediction error, and that is obviously visible here. When the T-shift, T-scale, and absorption scale parameters were empirically applied in the Phoenix platform, the IVIVC was improved and the prediction errors within the acceptable range as per the IVIVC guidance. However, when you look at the individual formulation, there was a bias for the slow release formulation. But when we tried to deconvolute dissolution using mechanistic method, there was a really robust correlation between in vitro dissolution and in vivo dissolution. And some of the reasons that can explain this is because, as Eshwari mentioned, diltiazem is a complex molecule. And some of the complexities is, for example, it is metabolized by CYP3A4 enzyme, and hence it is subjected to the gut wall metabolism. And the enzyme distribution in the gut wall is different. For example, there is a high amount of 3A4 in the proximal small intestine, but there is a negligible amount of enzyme in colon. So if your formulation is a slow release, maybe most of the absorption is happening from colon, where there is a minimal amount of 3A4, so there is going to be a less metabolism. But if your formulation is a fast release, where the absorption is mainly happening from proximal small intestine, then there is going to be a more gut wall metabolism. So there is a formulation dependent nonlinearity in the fraction absorbed, and which is going to become available in the systemic circulation. The additional complexity with diltiazem is that it inhibits its own metabolism. So the drug diltiazem, as well as its metabolite, both inhibits the enzyme CYP3A4. So basically, over a period of time, there is going to be a more and more inhibition of enzyme, and also increase in the exposure level. So this is a whole complexity involved with diltiazem, and when you count for all the disposition complexities, you would be able to estimate a purer dissolution data, and then you will be able to correlate well with in vitro property, that is dissolution. And the advantage with physiologically based method is that once the IVIVC is established, you can probably take it further, and there is also now a discussion going around that when the drug is known to accumulate, then probably the better IVIVC or better bioequivalence would be assessed at steady state rather than at single dose level. So once the IVIVC is developed in physiologically based method, you can try to run simulation for a steady state, and you can estimate a steady state level concentrations from a given formulation. As you can see, after 7 consecutive doses, the accumulation for fast release formulation is about 74%, as compared to only 46% for slow release formulation. That is because the slow release formulation is mainly getting absorbed from colon, and the auto inhibition of the metabolism is not going to affect much. But fast release formulation, which is getting absorbed from the proximal intestine, where over a period of time when the inhibition of the enzyme is happening, there is going to be a more accumulation of the drug. So probably the mechanistic approaches would allow you to explore beyond a conventional arena of IVIVC for a given formulation. Just to summarize the talk, the mechanistic model provides detailed understanding of the mechanisms involved, and probably explore what is happening in overall absorption, which is a composite profile of dissolution, transit, permeation, gut wall metabolism, and first pass metabolism. And then you can also identify the sensitivity parameters, or which are the critical parameters for your formulation. On the other hand, numerical methods are really a gold standard. And if the purpose is to follow the guideline and develop IVIVC, which is robust enough from the guideline perspective, probably the numerical methods are the best option, because they also provide a statistical and numerical rigor which are required for a regulatory submission. Other ways, as we saw in the Metoprolol example, that the both models can complement each other. You can also estimate fraction absorbed from mechanistic model and try to see whether it matches with numerical method. So basically, both of the method can serve as an additional validation for each of them. So with numerical method, you can estimate fraction absorbed. Try with mechanistic model and see whether you can explain if you get the same fraction absorbed. Otherwise, you can try to go back and see where the errors are in either of the model. So last but not the least, that the mechanistic model may be more helpful in formulation development or optimization, because it allows you to understand the mechanisms and what is the critical for your formulation. So that would help you to understand more while you are in the development and optimization phase. However, the numerical method may be more suitable for regulatory work, where you need a statistical and numerical rigor to validate and ascertain the model is robust enough to claim a bio waiver.